All right. So good morning, everyone. So today we have the honor of having Dr. Scott Blanchard give us a lecture. And so I'm just going to give you a little bit of a background of his upbringing, academic upbringing, and also his accomplishments. So he obtained a Bachelor's of Art in Economics at the University of California, Davis, and then went on to obtain a BS in Chemistry at the University of California, Santa Cruz, then a PhD in Biophysics at Stanford University, and then did a postdoctoral fellowship in applied physics at Stanford University. Um, his professional career after the postdoctoral work, he started as a professor at Cornell, Weill Cornell Medicine. This was in Ithaca, New York.
Nope,
And then
New
he
York City.
recently, New York City. Okay, sorry, correction. And then has since relocated to St. Jude's Children's Research Hospital in Memphis, Tennessee. So just a little bit of his research overview. So he has worked in translation mechanism, ribosome dynamics, and mechanism of antibiotic action, guanosine nucleotide exchange factor mediated catalysis of TUTS, GTP, AAT, RNA,
Mhm.
ternary complex formation and turnover, self-heating organic fluorophores, direct imaging of integral membrane protein dynamics, ribosomal RNA sequence variation from bacteria to mammals. Over the span of his career, he has participated in as part of an editorial board, participated in reviews of different grants, served as session chairs of conferences, participated in a number of grant reviews, scientific reviews, a number of accomplishments in terms of awards that he's received. 2019 Outstanding Mentoring and Teaching Award, Weill Cornell Medicine, 2019 Endowed Chair of Molecular Imaging, St. Jude Children's Research Hospital, 2024 University of Kansas Philip and Marjorie Newmark Lecture. He has, in addition to research, participated in formal teaching at St. Jude's. He has participated in the teaching of the Research Hospital Graduate School, lecture in genes to proteins. He has obtained 12 patents and other works in progress. He currently has active funds from an R1 in addition to other sources of funding. In terms of his publication record, 116 peer-reviewed primary articles in addition to peer-reviewed reviews and book chapters, and also non-peer-reviewed publication reports. His oral presentations exceed over 100. And so with that introduction, I'll turn over to our guest lecturer.
That's great, Arthur. Thank you very much. I debated whether to send you the full CV or the shortened CV, and I guess you took it upon yourself to give a plenary overview. Thank you very much. I'm not sure how many people I'm talking to. I'm kind of looking at a class that appears to be around 10. Is that about right?
Yeah, so we have a total of 13 students. But yeah, right now we have 10.
13 students. Great. Great. And this is a chemical biology and biomedical research course. So it's guest lectures for all the courses, all the classes, excuse me.
Yeah, the majority of the course. I began with a few lectures just as background for the things
Wonderful.
that yeah, will come.
That's fantastic. Again, thank you for the introduction, Arthur. For those of you who want to get a better understanding of the background and motivations for my research career, you can refer to the Biophysical Society website where I recorded a 10-minute lecture last year after receiving the Kinosita award for single molecule biophysics, and I spend 10 minutes and sort of give you the background of who were my mentors and who altered my career and the breakthroughs that I think we've made and where we're going. So I definitely encourage you to take a look at that 10-minute lecture again on the Biophysical Society website for the Kinosita award in single molecule biophysics. Okay, so what I'm going to try to do today is, again, in a much longer format, give you some sense of the motivations for my research career. Um, I was drawn to this field simply by, immersion. I tried it and I liked it, and and I liked it so much that I decided to pursue it as a career, and abandoning my dreams, early dreams of going to medical school, and the earlier dreams of going into finance. I want to start by acknowledging the many people that I've had a chance to interact with over the years, uh, and this is our current group right now. So this is here at St. Jude Children's Research Hospital, where I moved in 2019 to really pursue my dreams in research that had been sort of revealed during my early faculty career. So, but I've had the pleasure and distinguished honor of training about 20 PhD graduate students over the course of my career, from the rotation project through to the PhD thesis defense, and then placement in jobs. And this is really where it's at. The people who I work with really make everything happen. I should also mention that St. Jude Children's Research Hospital is a very wonderful place to work that has a very highly supportive environment that allows us to pursue the research we do. And they support me, the Blanchard lab down here, to basically run a single molecule imaging center, which I'll tell you about a little bit today and what we're trying to do in that center. And of course, over my 20 years of research, I've been supported very generously by the National Institutes of Health throughout, and the National Science Foundation intermittently, and that's been absolutely vital to our research. What I'm going to try to tell you today is not just the work of the current members of the lab. It's really the 24-year career that I've had. So the dream for my research. Oh, I don't have, my movies aren't embedded. I'm so sorry. The dream for my career really was to take static structural biology, cryo-electron microscopy structures or X-ray crystallography structures, and animate them as they occur in real time. And so this was the dream I had as a graduate student, was how can I add to the repertoire of tools that allow us to directly image biological systems by just simply watching one molecule, in this case the ribosome, do what it does as it works. And so, we knew from first principles and deductive biochemistry that the ribosome was a machine that underwent large-scale conformational changes during function. It moved, subunits have to come together on the messenger RNA. The tRNA molecules are flying in and flying out, and it's moving along the messenger RNA. And this system, in its obvious motions, really led me to think, okay, what we really need to do is use a tool where we can directly monitor the binding and association of factors and their movements relative to each other. And we dove into developing the tools to make this happen. And our thesis, if you will, was that the mechanism of protein synthesis would be very difficult to study in a test tube of billions of molecules, because you would be looking at, a heterogeneous system, both in terms of function, but also in terms of the physical composition that you were able to achieve let's say, in your reconstitution. And so at the end of it, you end up taking a measurement of these billions of molecules in the test tube, and you get a population average that could be very difficult to interpret, including telling you nothing's changing as a function of time, when really that's just the average of all of the individual behaviors. So our dream was to be able to put fluorophores on individual molecules and visualize where those fluorophores were, and then how they moved with respect to each other during function. Let's say, for example, a ribosome undergoing a conformational change as depicted here on this slide. And then, basically measure the duration of time that it spends in conformation A before converting to conformation B, then measuring the duration of time it spends in conformation B before converting back to conformation A, and then somehow watch these ordered sequence of events to directly reveal information about the mechanism of translation. And for those of you who are interested in learning more about single molecule methods, the foundations of the approach, where it came from, I would refer you to this brilliant research article, review article by Shimon Weiss, published in Science in 1999, which came out during the third year, of my PhD thesis research, and 3 years after the first observations were ever made of a single fluorophore on a biological molecule at room temperature. And so I just happened to be at the right place at the right time where this field was taking off. And so when I came to St. Jude, we were given the opportunity to make a movie that would sort of summarize what I just said. And this is a movie, if it plays, of the ribosome undergoing conformational changes during function, like that movie showed earlier, but this time with fluorophores placed in a manner that would allow us to visualize this process of movement through the messenger RNA. And so the idea is that we're going to figure out how to put the fluorophores on the ribosome where they need to go to inform on function. And then we're going to be able to watch ensembles of individual molecules and hopefully learn new things, including how to rescue dysfunctional molecules with antibiotics or small molecules. And so that little vignette, that whole movie can be seen on our website at St. Jude Children's Hospital Single Molecule Center. And that movie has a little bit more of an elaboration. But I wanted to impress upon you that it was based on real data. Those structures in that movie were real atomic resolution structures that we had solved in our lab. And we have really placed fluorophores where we showed them. But I will confess that the traces that we showed were simulated, though those weren't real. So as I said, this is the dream. And the question is, how do you realize a dream? Essentially, what you do is you strap on a backpack and you just start marching in the direction that you want to go. And what I'm summarizing here in this slide are all the different areas that we had to, basically master in order to reduce this dream to practice. And I'm not going to go through all these things in exquisite detail, but I just wanted to give you a sense that this is a really complex interdisciplinary approach where you need to know biology in order to ask the right questions and be able to isolate your biomolecules of interest. But you also need to build microscopes. You need to build software. You need to have expertise in sort of fluidics handling. As I'll tell you about today, we're learning how to build microarrays through spatial multiplexing. We have to have surface chemistry and we got to do prototyping. So we have a very interdisciplinary team from biology to practical engineering, but also math, where you have to use computer science to learn the language, if you will, of these molecules. And we'll come back to that later, I'm sure. So the bread and butter of our research really has distilled down to this instrument shown here, which is a prism-based wide field total internal reflection fluorescence microscope. And this is an enclosed system. It's environmentally protected. The lasers that we use are, under under darkness, and the mirrors and optics are protected from dust. And this is our workhorse, and we have three of these in our lab. And the schematic of what's inside that instrument are shown here in the center. And basically, we have four lasers collimated to then impinge on a sample, and through total internal reflection in a very small area inside this microfluidic device, we can illuminate molecules that are tethered near that point of total internal reflection, and then they emit light. And we collect that with an objective microscope lens, a magnifying objective microscope lens, and then funnel the information collected through optics to then parse the light of different colors onto different cameras. So we have one camera in this case for four different fluorophore colors, which are the fluorophores themselves are referred to as Psi2, Psi3, Psi5, and Psi7. And these are just jargon for chemical matter that allows us to visualize individual molecules. And so each one of these cameras is streaming data to memory. And this is what an individual image looks like from an individual camera, where you have tens of thousands of individual molecules tethered on your surface, which has been passivated such that they're not adhered, but they're just tethered. They're floating on top of it. So they're biologically functional. And when you zoom in on an area, you can see that you have point spread functions for individual fluorophores that you then collect computationally and extract for that information that we're looking for. Oops. This is a cartoon schematic of that total internal reflection point of incidence, where we're usually using a polyethylene glycol passivated surface that is optically transparent, and then that PEG is doped with biotin PEG, which allows us to use streptavidin, which is a tetramer, to then bridge our biological of interest containing a biotin with the surface. And that's the biotin streptavidin bridge I have depicted here. And this is a cartoon of a molecule that contains two different color fluorophores that talk to each other through fluorescence resonance energy transfer, which is a mechanism of that was sort of, I'd say, brought to bear on biological systems in the late 1950s, early 1960s by a gentleman named Lubert Stryer, who happened to be a Stanford faculty neighbor when I was a graduate student there. And this is a molecular scale ruler where you can excite the donor, and the excited fluorophore, in its effort to relax back down to the ground state, will transfer energy to a neighboring fluorophore if it's of the appropriate wavelength, called that's the acceptor fluorophore. And so here the donor is green and the acceptor is red. And if they're at the right distance from each other, you'll see fluorescence resonance energy transfer from green to red, and you'll get fluorescence out of the red fluorophore, even though you didn't directly excite it. So in that situation, the donor and acceptor will be undergoing anti-correlated fluorescence changes as the molecule changes conformation. And so that's what this slide shows you here is if you take this protein, which is a two-domain protein, and you put a donor on one domain and an acceptor on the other domain, and they're separated by about 70 angstroms, you can take that system and add at the KD of binding its small molecule substrate. And every time it binds the molecule, it will close around it. And then the distance between the fluorophores will reduce by about 15 angstroms. And that 15 angstrom conformational change, of course, will reverse when the leucine molecule, or in this case, falls off. So if you put it at the KD, you're just looking at binding and unbinding happening reversibly back and forth. And that's what this trace upstairs here is representing. So this is real experimental data of the donor and acceptor undergoing anti-correlated changes as a function of time. And just to go back to the details, this is what the fluorophores look like, the Psi3 fluorophore and the Psi5 fluorophore, which are the donor and acceptor, respectively. And again, these traces here are collected from those movies that are, obtained for donor only and acceptor only. We take those two movies and then we stitch them back together for each individual molecule because they're spatially localized in the same area. So we know that this donor correlates with this acceptor because they're spatially separated from everything else. So these are the types of data that we're collecting, and we're collecting in massive scale. And we've been building the tools to make this possible. And, from microscope development to fluorophore placement on our molecule of interest, to the analysis of these types of trace files, as they're called, that come from individual molecules to measure those dwell times and the number of conformations that it samples. So these microscopes require software development. I just wanted to highlight the fact that, we've spent 20 years building acquisition software to essentially run the microscope and tell the lasers when to turn on, tell the cameras when to stream, tell fluid when to run, and tell, and show the user the data in live format. And that's called Flash. And then once we obtain the single molecule traces from those tens of thousands of molecules in each experiment, we have to extract those data that I was referring to, which are the dwell times in the various FRET states. And the software that we've developed over the course of 20 years is called Spartan. And these tools are available on our Single Molecule Center website for people to download and use at their purpose. So I forgot to say, so the construction of the microscope and the construction of these software were absolutely vital to the success of my research laboratory over the years because, I come from a training in chemistry and physics, but I got my first job at a medical school. And at a medical school, there's not a lot of chemists and there's not a lot of physicists. So I really had to focus my early career on making these microscopes easily accessible and the data analysis packages easily interpretable. So these were vital to my career and the success that we've had. The second major breakthrough relates to the fluorophore engineering, as Arthur introduced. And, these Psi3 and Psi5 fluorophores, these are commercially available species that you buy. At the time, it was GE Healthcare that was selling the fluorophores. And it turned out that during my graduate career, the fluorophores were back ordered and I couldn't order the fluorophores. And I realized that I can't be dependent on a company that sells fluorophores, sporadically. So I took it upon myself to start learning how to synthesize fluorophores myself to give me a little bit more flexibility. And through the course of the synthesis and the analysis, I realized that organic fluorophores are fundamentally flawed. And they're fundamentally flawed in the following graphical way up here in the upper right, which is you collect photon emission over time, and that photon emission is very highly varied, right? So it's dancing all over the place. And the faster the time resolution you take, the more it's dancing. And moreover, it dances and then it terminates. And this termination of fluorescence is called photobleaching. And so what you end up getting in a movie is average number of photons emitted per frame, and then a total duration of fluorescence that multiply together to give you your total photon budget. And your total photon budget essentially equates to how much information do you have to extract what you need. And the fluorophores that were available commercially were abysmal. This is actually a very good trace here, but it's giving, in this case, it's giving out 600 photons per frame, and it's lasting for about 55 seconds. That's reasonable. But we, through our research, realized that we could fix these fluorophores by attaching a molecule to them, a very special molecule called cyclooctatetraene. And that attachment of cyclooctatetraene to any organic fluorophore would improve its performance from this type of behavior to this type of behavior. So it goes from 500 or 600 photons to 5,000 to 6,000 photons per frame. And the variance in the photon emission goes from very noisy to very tight. And it lasts about also 10 times longer. So we're getting a massive increase in our total photon budget, and the quality of the data is much higher. So this really took us from a situation where we were doing very complex analysis of real biological data mixed with photophysical noise to analyzing more pure biological data. So these properties of fluorophores to exhibit photophysics is sort of beyond the scope of this lecture. But suffice to say that if you remember your chemistry, the highest molecular orbital has 2 electrons in it of antiparallel spin. When upon excitation, 1 electron goes to the lowest unoccupied molecular orbital. And then if you're lucky, it'll emit a photon as it relaxes back down to the highest occupied molecular orbital. But it turns out that the photophysics arises not from the process of going up and down and up and down, but it arises from the 1 in 100 times that the electron forgets its spin when it gets excited, and then it's trapped because you can't bring 2 electrons back into the same molecular orbital with the same spin. So that's called Hund's rule. And so it gets trapped in this higher order excited state, and it's not fluorescing. So every imaging frame, it's undergoing multiple excursions to these trapped dark states, and that's what's giving rise to the noise, because that's probabilistic. Anyhow, this state that we're talking about, this trapped dark state, is called the triplet state. And so that's shown here. You have a ground state fluorophore that's excited into its excited state. And what you want it to do is emit a photon from that state and return back to the ground state. But that spin flip puts you in a triplet state that's dark. And that dark triplet state then reacts with molecular oxygen, and it leads to photodestruction of your fluorophore or damage to your environment. And so our method of attaching this cyclooctatetraene molecule actually quenches the triplet state and returns it back down to the ground state directly. And that's shown here through these direct measurements of the triplet state lifetime of an organic fluorophore such as Cy5, and on the order of 60 to 100 microseconds. When you attach COT to the fluorophore, you can collapse that down by almost 100-fold. And that 100-fold shortening of the triplet state is what gives rise to the better fluorescence behavior. And this was all carried out by a graduate student in the lab who's was very prolific, named Qinzi Zheng. So back to the practicalities. Here's what a parent fluorophore looks like in a movie: obscure and dark and blinky. And here's what these fluorophores linked to COT look like, which the field has decided to call self-healing fluorophores. And these self-healing fluorophores are just bright as you can imagine, and they last for a very long time. But more importantly, they when you turn up the laser power, they don't saturate because they go almost all of it to triplet states. They just get brighter and brighter and brighter. And so you get faster and faster time resolution data such that you can start measuring biological processes that are happening in the millisecond to sub-millisecond time scale. So these microscopes and the fluorophores are really the foundation of our work. And we're at St. Jude working to extend these technologies of self-healing to a diverse class of fluorophores across spanning the visible spectrum, where we can somehow bring these to bear on it, whether it be in vitro biochemistry or immunofluorescence or live cell imaging. We're really hoping to bring these to the imaging community because they are really much better at giving you biological information and they're less toxic. So those are the three foundations of my lab that give rise to our aggressive pursuit of more complex biological problems. So I'm not going to go into every biological system that we've had the privilege of investigating over the years through our collaborations, but suffice to say, I was the person, I think, that really wanted to take the foundations of single molecule imaging and bring them to bear on complex biological problems that were of medical relevance. So my interests began in graduate school with the bacterial ribosome, as I've sort of alluded to. And my interest there was antibiotic mechanisms of action. How do antibiotics work? My thought was if I could understand how known antibiotics work, I would be able to then learn something about the principles of regulation. Um, but during the course of my career, I've extended that to the investigations of human ribosomes, which are potentially anti-cancer targets, as well as a host of other, diverse biological systems spanning from integral membrane proteins to ribozymes and riboswitches, and all of those works are published with experts in these individual areas. And my goal was to collaborate with them and then use single molecule imaging to teach them something about the dynamism of their system that would reveal something that they didn't know. And in every case, because we were successful publishing, we were able to demonstrate that this imaging approach that I've just sort of laid the foundations for is able to tell experts something that they didn't know about their system. So let me just take a second and talk about one of our more recent collaborative studies on the cystic fibrosis transmembrane receptor, which is a protein, an integral membrane protein, which transports chloride across epithelial cells. And malfunction or dysfunction of that protein leads to cystic fibrosis. And so we decided to set out with a collaborator named Ju Chen to try to put fluorophores on this system and learn the mechanism of function. And I'm just summarizing that here as a way of sort of communicating to you that from these individual traces of individual molecules, we're learning more than just the dwell time of conformation A and conformation B. The goal of our platform is to actually delineate a kinetic mechanism of function and an order and timing of the events. So how does this cystic fibrosis transmembrane receptor bind ATP, undergo conformational changes as a consequence of binding, and then allosterically communicate that gate opening and time that somehow with ATP hydrolysis. So this paper is published, and you can look and see how we deduce this mechanism from our single molecule studies. But it was a collaboration where we're taking data from single molecule research and we're integrating it with biochemistry measurements of ATP hydrolysis, and we're making measurements of of ion channel conductance with electrophysiology. So it's three different methods that come together with the inclusion of cryo-EM, and we land on this very complete model where this cystic fibrosis transmembrane receptor is basically dancing between all of these conformations stochastically, but because there's energy expenditure, it has a net effect of going in a cycle. So this is an integration of single molecule FRET, ATP hydrolysis measurements, and electrophysiology, and we land on a very minimal kinetic model that says there has to be at least 8 different conformational states of this receptor. And so that's sort of where we begin, right? So we take our measurements, we establish a biochemical framework for understanding function, and then inside that framework, we start testing whether or not it holds up when we add mutations or drugs or other types of perturbations. And so the kinetic model is not the end, it's the beginning. And of course, wouldn't you like to have high resolution atomic structures of each of these individual states so that you could then have three-dimensional pictures for all of these different predictions? And so that's what we're pursuing through our collaborations. So hopefully in this early vignette, I've convinced you that single molecule biophysics provides an approach to measuring conformational changes relevant to function in diverse biological systems. And with that, I think that I can begin the deeper dive into where this field is going. And our excitement about potentially using this approach for high throughput drug discovery. And so I'll take pause and let questions happen before I go on.
Does anyone have a question?
Is that a no?
Yeah.
There's a
Hi.
question. Can you hear him?
Okay.
Can you hear me?
Yes.
Okay, perfect. Okay, I just had a quick question. I think this might be a little bit basic, but I was still wondering, like, how do you decide where to place the fluorophores on the molecules, and how do you assess like possible effects on binding, affinity, and kinetics? Comparing
Amazing.
the one that you're.
That's a great question, and that tells me that I explained things clear enough that you understood. So that is the $1 million question. How do you put the fluorophores somewhere that's informative on function? And so I definitely glossed over that. The way we generally do this is these very, very important biological molecules are usually the targets of many structural biology labs that are trying to solve atomic resolution structures in order to understand function. And invariably, in this modern era of cryo-electron microscopy, they will report multiple structures. And those multiple structures then, obviously are either one's an artifact and one's reality, or more, as we now understand, they're converting between those structures. So those conformational changes from static structures can be inferred, and those are what we use to place our fluorophores at informative sites. But generally speaking, those are our starting point. We also use molecular dynamic simulations. We use AlphaFold, and we use other types of modern tools to understand those conformational changes. But nowadays, we have sort of a system where if somebody comes to us with a naive biological molecule, we should be able to pinpoint where to label it. Great question.
I think that's the only question for now.
Great. So what I wanted to spend some time doing is sort of a chemical biology excursion into how do we take this method, which we've already demonstrated is reasonably powerful. How do we make it even more powerful by increasing experimental throughput and enabling even greater sensitivity than we already have. Um, so let's start with the very practical, which is I train people to learn how to do this approach. And they obviously go through a process of learning where they come in and they're completely naive and they're making mistakes and they're taking data on a Monday and it's not looking like the data on Tuesday. And sometimes for a paper, you think you're done with the paper, but the reviewer asks you for more data. And then you go back and you take more data and you're like, dang, it doesn't look like what I took before. So all of the things I've discussed were really about making this robust and reproducible. But in those investigations of those many systems, we realized that, these measurements are extraordinarily sensitive and, very subtle changes in temperature even um, cause problems. So that black box that we have is temperature controlled because in New York City, in the winter and in the summer, you can't keep it, the open microscope, within 2 to 3 degrees. So if you take data in the winter and you take data in the summer, it's got different rates of dynamics. So we really had to build that box. And then this is the next method to really help us compare two systems head to head where we know we have the exact same um, conditions. So we want to make an approach that gives robust comparative studies under identical conditions. We want it to be applicable to a broad range of biological systems, and we want it to be compatible with both steady state measurements of the types I've already shown you, but also pre-steady state measurements where you subject your system to a stimulus rapidly. So that's called pre-steady state or stop flow. So the way we approached that was taking our existing platforms and our existing cameras, and then using micro contact printing to provide specificity for different molecules to localize to different areas. And this is what I'm going to try to walk you through. So micro contact printing is a method that's very well established. And you basically take a fabricated pin of a very small dimension, about 100 microns, you drop it into a 96 or 384 well plate, and then you pick up about 50 picoliters, and then you place it onto a passivated quartz surface that we were already, the tines we were already using. And if you then rinse the sample and then drop another biomolecule in a different area, you now have spatial separation in a defined volume, different types of molecules. So what we're doing to encode the binding of different types of molecules is we're printing oligonucleotides called anchors of a defined sequence at pre-complex with streptavidin. So we're printing microarrays of these DNAs of specific sequence. And then if we add a biological molecule with a complementary strand to that anchor sequence, it will localize and hybridize to that region. And then through a technology called tow-mediated strand exchange, you can add a switch oligonucleotide, shown here in gray, and it will bind to your biological molecule and it will strip it away from the surface by complementary hybridization that is greater thermodynamic stability than the anchor that was holding it to the surface. So if you take, 4 biological molecules of interest with 4 different tether strands sequences, and you've printed 4 different anchor sequences, what you're hoping for is that they'll localize to these individual spots, and then you have 4 different biological molecules in one field of view. So this field of view is shown here in green. One of the, maybe nuances that might be of interest to some of you in the audience is, how do you have even illumination across this field of view? And that's done by something called top hat illumination. And basically, the top hat illumination takes a Gaussian beam and it makes it square. And so that's shown here. This is real experimental data of a Gaussian beam that we've done flat top illumination to make square. And what that does is it takes every molecule in your field of view and gives it the exact same excitation profile. So you get the exact same emission profiles, and your very noisy data from a Gaussian distribution will turn into a very narrow, tight distribution of intensities. And so this is actually very simple, reasonably established technologies for using optics to shape light. So now that we have microcontact printed, or I've hopefully convinced you that we can microcontact print molecules, we can do this in large arrays inside of microfluidic devices that have inlet and outlet ports. And now if we encapsulate that patterned microarray in this microfluidic device with inlet and outlet ports, we can connect this to microfluidic control systems, much like an HPLC. And so here we have arrays of molecules and we have fluidics controlled by syringe pumps. And then this is all under control of that flash software I mentioned earlier. So this is the foundation of this paper we published in just this year that describes this parallelization of single molecule imaging. So just to convince you that this method of microfluidics is got capabilities for pre-steady state, I'm just showing you how fast we can mix one buffer into another buffer, those immobilized molecules. And the answer is about it's about 50 to 100 milliseconds is sort of our sweet spot range. So you can take a molecule in solution A and you can exchange it in 100 milliseconds or less to solution B, and that's really powerful for adding stimulus, etc. And another aspect of this system is that you can do this repeatedly. You can take solution A and move it to solution B and then rinse it back to solution A. And you can do this over and over and over again to do repeats of your experiment. And so this is an example of 18 repeats and all overlaid on top of each other with the error bar in mixing, shown here in gray. So this is highly reproducible fluid control of a single molecule scale. So let's put it to work. So this is that amino acid binding protein I was talking about before, this model system. And here is us adding leucine and then taking data for half a second and then rinsing out leucine. And this is 18 injections. And you're showing very rapid, very reproducible binding and release of this leucine molecule because it has very low affinity, micromolar affinity. So you can do this all over again as many times as you want, as long as your biological molecule tethered to the surface doesn't lose its function. So this is our workhorse model system, but it, of course, can be applied to many things. So I'd like to walk you through 3 different examples of diverse biological systems that this is amenable to. I'm going to start with the simplest, DNA oligonucleotides. So this is that tether approach that we're talking about, and we're going to take 4 different DNA samples that are labeled with donor and acceptor fluorophore at different spatial separations. So there are 4 different constant FRET values that we expect from these 4 samples. And if our method is correct, we should be able to get each quadrant will have 1 FRET value. So, but we learned here in this early experiment the power of this hybridization and switch strand epitaxial removal, because you try your best to estimate the concentrations of your biological samples with, say, a UV trace, but you're never going to be exactly right. And so you end up with situations where one molecule is very high density and the other three are not high enough or just right. So you really want to use those switch oligonucleotides to even out the concentrations immobilized on the surface. So you have a perfect distribution of molecules in each one of those areas. And this is the first evidence that we have control over the specificity of binding to each of these regions. And so what you see here is we overshot the concentration of sample A, and then we can bring it back down to the appropriate concentration with a switch oligonucleotide, rinse out the switch oligonucleotide, and then stabilize its surface density. Mm-hmm. Now all four are about the appropriate density. But let's say, for example, you got something wrong and you sample D, you're like, oh, I added the wrong sample. You can strip it off and add another sample and put it back on. And then at the end of your experiment, you can strip them all away and then you can start over. So this switch oligonucleotide technology which is summarized in a paper that we published I don't know when that was, called ERASE. And this ERASE technology is really powerful. So that's summarized here, where we have 4 different fretting oligonucleotides. You mix them together, you add them all at once to your pattern microarray, and boop, they go to where they're supposed to go, annealing at room temperature. So this method of finding the appropriate anchor tether pair, I'm sort of skipping over, but there's computational programs for you to do this, and you can do this at scale. There's lots of oligonucleotides that you can program to give you this orthogonality. And so, as I said, and as I demonstrated, these are very tight, narrow distributions that have essentially no crosstalk contamination with other binding of other oligos. And so these are lower estimates, 95 to 99% specificity. And so this is exactly what we need to go forward and start doing measurements with more confidence. So the first experiment that we did, besides that fretting oligonucleotide, is we said, let's take a broad category of commercially available fluorophores and just examine their performance under one environment. And so these are 4 green fluorophores and 4 red fluorophores, which we're mixing now 8 samples together, and we're getting 2 molecules in each one of the spots, 1 red, 1 green in each spot. And then we're going to cycle the excitation between green and red, and we're going to examine the green, then we're going to turn off the green, and we're going to examine the red, and this is called alternating laser excitation. And we can get that intensity and signal to noise and photon budget out, and that's summarized here. The results don't matter, but we can do 8 fluorophores performance simultaneously in this approach, and that's really vital when you're synthesizing fluorophores and trying to make better fluorophores for biological research. So this examination of fluorophore performance is absolutely critical. So let me move forward on to biological molecule example number 2, which is this small protein that's also a little bit like that leucine binding protein, but this is a molecule that's very vital to cellular physiology called beta-arrestin. And so with this approach, we're going to try to take a beta-arrestin molecule labeled with donor and acceptor fluorophore, and we're going to immobilize it by a DNA oligo tagged antibody. And that antibody binds a genetically encoded N-terminal affinity tag called a strep tag. And so you can make your arrestin molecules labeled, you add them individually to the antibody with its own tether linked oligonucleotide, and you can do this parallelization again. So let me spend a moment. Arrestin molecules are involved in GPCR signaling. G protein coupled receptors are in the integral membrane protein that reside in your extracellular membrane or your internal membranes, and they mediate the transmission of information across those lipid bilayers. And these are involved in almost every aspect of physiology, from vision to temperature sensing to blood pressure regulation. Anything you can imagine, GPCRs are at the center of it. And arrestins are molecules that terminate signaling through GPCRs by binding to them once they become phosphorylated and then bringing them into internal compartments for endocytosis and ultimately degradation. So these arrestin molecules are very, very special place in biology. And it's been realized more recently that they also bind to phosphorylated GPCRs and then mediate signaling on their own. So they can actually carry out coupling to other systems all by themselves, and the diversity of systems that they couple to is expanding. So we were interested in building on a collaboration that we had started with Bob Lefkowitz, who is the godfather of GPCR biology. He cloned the first GPCR, and he discovered the arrestin molecule. And so we decided to collaborate with him to ask if we could measure conformational changes in arrestin in response to binding phosphorylated mimics of the GPCR molecule. And that mimic I'm going to refer to is called V2RPP. And this is just a peptide that's phosphorylated in a specific pattern, and it's been determined by the field to be a great binder to the arrestin molecule. And so this paper we published in 2022 with his group showed that when you add this V2RPP peptide to a donor and acceptor labeled arrestin in a manner I've shown schematically here, when it binds, it displaces an autoinhibitory tail on the C terminus, and then that gives rise to a change in fret from high fret to low fret. And this is an example, a trace that was published in that paper, where we can take arrestin, we can add a little bit of V2RPP, and then it starts going back and forth, and then we can add more V2RPP, and now it's stuck in the open low fret configuration, and then we can wash out the peptide and it comes right back. So this was our foundation, but what we really wanted to do in collaboration with Bob was get deeper into this process, where people had reported that this arrestin molecule, in addition to having the release of this autoinhibitory C-tail, it also underwent rotation and it underwent other dynamics in different regions of the molecule. So what we set out to do was label it so that we could see all of those distinct conformational changes in different molecules. So we label one molecule to measure C-tail release. We label another molecule to measure this finger loop dynamics. And we label another molecule to measure rotation, all done through, basically predictive structural biology based on these static structures that had been solved. And so we immobilized those samples. We included a fourth sample, so we have two structural perspectives on the C-tail release. And we immobilized those in our patterned array, as you would already imagine. And then we subjected this to some V2RPP stop flow measurements to ask, how fast does V2RPP bind to each of these different constructs? And through a series of concentration-dependent analyses, we can get the on rate, and through the rinsing, we can get the off rate. And so I'm really summarizing a lot of work here, but we were able to quantify the on and off rates of V2RPP from these different constructs and start quantifying those head to head because they're under identical conditions. And so I'm going to come back to the question that was asked earlier, but the summary of that work is that these data suggest to us from a kinetic perspective that the finger loop moves first, then the C tail releases, and then the C domain rotates with respect to the N domain. So this is the early signs that there's actually an ordered sequence to those conformational changes that have been described through static structures. And so this is where you begin to start exploring whether this is real, and what biological implications does an ordered sequence of events have. And so I just wanted to give an acknowledgement to the question earlier. In addition to the informative sites of labeling, you also have to make sure your labeling didn't screw anything up. And so this was a story that's published in the paper where we realized that our site of labeling on the finger loop was actually causing at least some aspect of the perturbation. But even after we fixed it, it was still showing this faster rate of conformational change in response to V2RPP. So the summary is that this system undergoes an ordered sequence of conformational changes where the finger loop moves first, C tail then releases, and then the rotation happens. And so what are the implications? So, well, there's 4 different arrestin subtypes that are present in nature. And we want to ask, is that true for all the arrestins? What happens when you inhibit one conformational change and test what that does in cells? I also told you that this was what we see for just the V2RPP peptide, but it turns out that every receptor has a different phosphotail, and even the same receptor can have different phosphorylation marks, and those distinctions actually have very important physiological consequences. So we're very excited to take this forward and do the necessary experiments to really validate this model, and that's going to come through more complicated experiments that I'll save for later. Does anybody have any questions before I move on?
Any questions? Yeah, no questions at the moment.
Great. So with these successes of the DNA and the beta arrestin, which I should mention is generalizable to this immobilization strategy is generalizable to any protein with a tag, an affinity tag on it. We thought, let's see if we can extend this to immobilizing RNA, molecules that have RNA in them. And so we turned to the bacterial ribosome as our bread and butter. And basically, what we're trying to do here is take a ribosome that has a given messenger RNA associated with it, and then use this anchor technology again to get that specificity. And so I'm going to try to walk you through a couple vignettes, short ones, that hopefully indicate that this is viable. So I want to impress upon you just real briefly, who cares about ribosomes? Haven't we figured out the mechanism of translation already? And the reason I want to spend a minute on it is because I think ribosomes are still the most important thing for us to study. And that's partially because they're found in such high abundance. And here's just an example of 2 tomographic images of, showing sort of what the estimated concentration of ribosomes are in cells. And I just want to say that in both bacteria and mammals, when you crack these things open, contrary to the cartoons that are shown on textbooks or Twitter or whatever, there's ribosomes just about everywhere. And that it is a lot of the cellular energy, more than half, is spent making ribosomes and making proteins with those ribosomes. So if you're at a cancer biology center, as I am, and you're trying to figure out how to target cancer, I think, nature tells us that this is a great drug target because 60% of all antibiotics that are known target bacterial ribosomes. So anyhow, this is a process that is very highly conserved between species, and it's just an amazingly important mechanism that many people overlook. So our long-standing motivations in this are to try to develop methods, single molecule methods, that allow us to look at the elongation mechanism, the mechanism by which proteins are made from individual messenger RNAs in both bacteria and mammals, to compare and contrast their mechanisms, to explore the question, how do antibiotics that target the bacterial ribosome not target the human ribosome if they're so conserved? And this is a pretty open question. And then on top of that question is, maybe can we target the human translation mechanism for therapeutic purpose? So there's got basic biology questions. It's got potentially medically important implications. This has been a focus. And just to remind everybody what the elongation mechanism is, it's a ribosome positioned on a messenger RNA with an mRNA codon unpaired with anything in its leading edge or aminoacyl site. This A site can then bind a ternary complex of tRNA, amino-acylated tRNA, in complex with a chaperone called EFTU bound to GTP. And all of the ternary complexes in the cell are binding and unbinding to this vacant A site until the right ternary complex is found that is appropriate for that messenger RNA codon. And then the magic happens where conformational changes occur. EFTU is stimulated, or this GTPase is stimulated to hydrolyze GTP and then fall off of the ribosome, allowing the tRNA to enter and undergo peptide bond formation, where the nascent chain linked to the tRNA already present is transferred to the newly incorporated tRNA. And then once that newly incorporated tRNAs got the nascent chain attached to it, then a series of other magic happens where conformational changes occur, leading to another GTPase binding, a different GTPase binding to the same site to catalyze the tRNAs and the messenger RNAs to move, to vacate the A site for another round of ternary complex binding and selection. And so this cycle happens hundreds to thousands of times for every single messenger RNA. And this is where a single molecule has really shown its light. To add further emphasis to why this is important, the elongation mechanism has been, is targeted by many antibiotics used clinically, and those have been characterized structurally with static representations of where those molecules bind with respect to this tRNA messenger RNA pair that has to take place. And they're all right on top of it, really. And most of them are not binding competitively. Most of them are binding in the region and somehow affecting the process. And of course, that binding has to somehow affect how the ribosome decodes or interprets the genetic code, if you will. And so many of these drugs are categorized as miscoding agents, where they bind the ribosome and the ribosome gets lazy and it picks the wrong ternary complex instead of the right one. And so those errors lead to catastrophe in protein synthesis, and then you stop making proteins and the cells die. So we've been trying to study this process of elongation with single molecule work for a long time, and I'm just showing you one method for doing so. And this is basically, you put a donor fluorophore on the tRNA that's already in the ribosome, you assemble this in vitro, and you put a messenger RNA of defined sequence in it that presents a given codon in the A site. And then we're going to use that messenger RNA to hybridize to our spot, our anchor tether oligonucleotide. And then after we've got them tethered, we're going to deliver by stop flow a ternary complex that has the appropriate or inappropriate ternary tRNA with an acceptor fluorophore on it. And when it binds, you go from 0 fret to some fret. And then when conformational changes occur, you go from some fret to higher fret. And then when peptide bond formation occurs, you see higher fret again. So you see this stepwise increase in fret as this tRNA binds and incorporates. And this is what the data look like, the primary data look like on the microscope. You inject the ternary complex to the cognate, that is cognate to the messenger RNA codon of interest, and you see binding followed by a conformational change, followed by another conformational change, followed by photobleaching. And so when you take these individual molecules and you stack them up on top of each other, synchronizing each trace to the first observation of fret, then you get a very clear picture that all the tRNAs are going through this projection of 0 fret, you bind a low fret state, you go to an intermediate fret state, and then you go to a high fret state. And that whole process, that genetic code selection, is happening in less than 50 milliseconds. So, just like a blink of an eye, that all of those conformational changes are taking place. So with this foundation, we wanted to look at miscoding of antibiotics. And so this is done by taking this parallelization approach and putting the two distinct cognate tRNAs for a given cognate messenger RNA codons for a given tRNA, in this case, the phenylalanine tRNA, which decodes UUU and UUC codons, UUC codons. So these are both cognate. And then at the same time, we want to probe near cognate, meaning it's close, but it's not the right codon. So these are CUC and UCC. So these are, generally speaking, single hydrogen bond changes in the messenger RNA. Everything else is the same. And so we set this up, and then we do the stop flow delivery, and we ask, what do the data look like? And the first thing we realize when we quantify these data, focus on the gray here for a second, that cognate tRNAs accommodate into this high fret state very quickly, on the order of, 2 to 3 per second in this measurement. And that's dependent, of course, on the concentration of ternary complex. Whereas near cognates are almost in indetectable amounts of incorporation. And that's what you want for a ribosome. You don't want them to, incorporate the wrong ones. You want them to incorporate the right ones. And then, when we add the antibiotic streptomycin, what's remarkable is that the cognate tRNA selection slows down dramatically, while the near cognate speeds up. And so this is very striking, is cognate tRNA selection slows down, near cognate shows up. So we wanted to analyze this and get into this a little deeper. Which one of these steps is affected? And I'm summarizing in quick form here the results. So what I want you to know is this is the presentation of the data for the two cognates and the two near cognates in the exact same experiment with one concentration of ternary complex. And all the data is synchronized to the first observation of fret. And so what you see is that the ternary complex binds to low fret, goes through intermediate fret, high fret for both cognate systems, although you could argue that those are occurring at slightly different rates. And then the near cognate is binding. It's trying to undergo conformational change, but it's falling off. And in the presence of streptomycin, what you see is the cognate systems don't slow down at all. They stay almost, the progression through those fret states, if anything, is faster. Whereas the near cognate tRNAs, now they get accepted very robustly. So to make a long story short, we analyze all these kinetics, and sure enough, after the tRNA binds, streptomycin makes every step faster for both cognate and for near cognate. And what streptomycin actually does is it binds to the ribosome and it prevents it from binding ternary complex. So it does cause errors, but it slows down the binding of ternary complex by about an order of magnitude. And that is probably the mechanism, the primary mechanism of inhibition of cell growth. And then the consequences of miscoding are on top of that. So this is a very nice vignette that I hope convinces you that this approach is amenable to analyzing RNA tethering, and it's very informative to examining aspects of genetic code fidelity, which we think is just an amazing opportunity for us to build upon and to start looking at, is this general for other antibiotics that target the ribosome? What about other ternary complexes? Everything I showed you was just tRNA phi. There's 50 different tRNAs in the cell that incorporate into the ribosome. Nobody's ever looked at whether aminoglycosides or other drugs affect all decoding processes the same. So we're really positioned to go in this direction. I wanted to pause there and ask questions and ask how much time I have left with you, because I'd like to walk you through one more vignette.
Okay, yeah, we have just a few more minutes left.
Okay. I'll go very, very quickly. If you want me to stop here, I can.
You can go through the. It just has to be pretty efficient. Yeah.
Yep, I'll go fast.
Sure.
So, in our studies of ribosomes, I've kind of alluded to the fact that this single hydrogen bond changes in the messenger RNA codon is the fundamental basis of fidelity. The ribosome is able to discriminate based on single nucleotide polymorphisms in the messenger RNA codon. And that's, basically the ribosome is making decisions worth whether a tRNA is correct or incorrect in like 1 to 5 milliseconds. It's lightning fast. But we realized in this view that if single nucleotide polymorphisms are the fundamental basis of the genetic code, what are the implications of the fact that the 7 different operons for ribosomes in E. coli are all different at the level of sequence? What does it mean that the E. coli genome encodes these different sequences of ribosomes? And so these single nucleotide polymorphisms are distributed around the periphery of the ribosome. And so everybody for the longest time was like, these don't matter. They're distal from the functional sites for tRNA selection. But we thought we would dive in. And I just want to summarize this paper, this very exciting paper, performed by a graduate student, Chad Curlow, in collaboration with others in the lab, where he showed that under physiological stress, the 7 different operons in E. coli actually, they all go down in expression, as you would expect for nutrient limitation, but they exhibit a change in relative expression when you measure that pool exactly. So some don't change in relative expression, some go down at a relative basis, and some go up. So what this data says is that E. coli doesn't make more ribosomes equally from each individual operon. It changes the pool of variants that are present in the cell. And so we wanted to ask if that's important. I'm going to skip. So the first thing we noted is that this sequence variation is conserved in bacteria that have this feast of famine lifestyle that E. coli do, which is in the animal and then on the ground. So that's feast in the animal, famine on the ground. So why would it be conserved? So we just set up an experiment to make E. coli with only one type of operon expressed from a plasmid. And these operons are exactly identical, but we've taken the single nucleotide polymorphisms in the operon that goes up in nutrient limitation, and we've added it to the operon that goes down. So now we have 10 SNPs. These SNPs are conserved, and we just wanted to ask, what is the impact on gene expression? And this is what we showed, is that 20% of the transcriptome changed when you compared these two cells that are absolutely isogenic, except for the 10 single nucleotide polymorphisms in the ribosome. And the gene expression wasn't just randomly changed, it was the gene expression responsible for the stress response. If you express the variants, these 10 SNPs, you got an exacerbated stress response. So that says that E. coli upregulate this operon because it potentiates the stress response. Closing now. How are these things different? How are these cells different? Or these ribosomes different physically? And this was a hint. We made these two strains, as I said, with the SNPs or no SNPs, and we then subjected them to a biolog phenotypic array where we just said, under what conditions does one strain grow faster than the other strain? And we arrived at the realization that they exhibited different sensitivities to tetracycline. If you had the SNPs, you were resistant to tetracycline. If you didn't have the SNPs, you were resistant to oxytetracycline. Tetracyclines bind directly to the ribosome in the same domain as the position of variants, but not near. So to summarize, we use the parallelization approach to then ask, can we recapitulate these phenotypic drug sensitivities and reduce it to the physical ribosome itself? And that's what I show here on this slide in very quick summary, is that when we analyze these B and H ribosomes, I think I missed, there we go. When we analyze these B and H ribosomes, the wild type and the variant ribosome, side by side, we can measure kinetic differences in response to, in tRNA selection, in response to these drugs. So the drug sensitivities that are differing between these two ribosomes are imbued by the variants encoded in the sequence of the ribosome. So something about the dynamism of the ribosomes is different. And we think that may be one of the reasons why you have changes in gene expression. So with that, I want to say the parallelization method we described is a powerful approach to monitoring diverse biological systems, and it offers a means for us to absolutely know the identity of the molecule you're looking at a priori. There are other methods that are being advanced where you try to figure out what the molecule we're looking at after the experiment's done, but this is a priori. And our method is, applicable to diverse systems, as I've shown you. It's applicable to pre-steady state measurements and very high signal to noise, very high time resolution things. So we're very excited about where this will take us on the road ahead. And with that, I'll just acknowledge again the lab, the support from St. Jude and the NIH. These people make this work, and it's one of the distinguished honors I have is to train these people. I also want to make one more mention that St. Jude Single Molecule Imaging Center is just one of many centers here that make this a great training environment. We've made use of the Single Molecule Center and the Cryo-Electron Microscopy Center, our own research, but there's also, many other electron tomography has now come. We have computational biology, NMR, crystallography, and mass spectrometry. And all of these centers and the entire St. Jude infrastructure is growing and expanding. So with that, I'll stop and we have time for questions. Thank you.
All right, so because of time, we're going to just give a round of applause for our speaker. Scott, thank you so much. We really appreciate it.
Wonderful. Thank you very much.
All right, then have a great rest of your day.
Okay, thank you.
All right, then take care.